Hi, everyone, and thank you. My name is David, and today I'll be discussing a theoretical model and an interactive app that we developed to explore how chromosome inversions might couple with ecological and mating bias multilocus genotypes to promote reproductive isolation in sympatric speciation. And so our group is looking to develop a complete mechanism to show how sympatric speciation can occur. And one of the questions we'd like to answer is how can we overcome the homogenizing effects of gene flow? And so, since sympatric speciation relies on like mechanisms that promote differential assortment of adaptive traits, it's important to understand how these mechanisms, what kind of mechanisms can play a role in that. And so, one of those mechanisms we believe is that can aid in this is chromosome inversions, and we think they'd act as a late stage adaptive barrier mechanism. We say late stage because there needs to be some non-random assortment of advantageous alleles already present in a population. This would make it easier for a chromosomal inversion to capture these advantageous combinations, and adaptive in the sense that they need to preserve the beneficial allele combinations. That's only really advantageous if hybrids are less fit. So without this hybrid disadvantage, the chromosomal inversion really has no benefit and wouldn't be able to invade. Um, so we believe the chromosomal inversions can form supergenes that will reduce recombination and maintain the linkage between these adaptive traits. So this work that we've done, it builds off of a previous model. So in our previous model, we had a 3 ecological locus model with 1 mating bias locus, and it did not allow for viable hybrids. In this model, since most phenotypic traits are polygenic in nature, we wanted to understand the dynamics of these polygenetic traits, how the dynamic of these polygenic traits are sorted is like integral to the process of sympatric speciation. So yeah, we allow for multiple ecological loci and multiple mating bias loci. And we also allowed introduced viable hybrids into our model, and then chromosomal inversions can capture any combination of ecological or mating bias alleles. So we have a picture of the general algorithm that was developed by Dr. Lin and Curtis. And then on the right, we have our mathematical model that was developed by Curtis. Okay, so I'll go ahead and start with the ecological model. So the way the simulation works is each individual will have multiple locus ecological genotypes, and they'll have either an A or a B allele. And then the ecological genotype will determine which niche each individual is assigned to. And then the individuals in the ecological model carry 1 mating bias allele, either an X or a Y value. And then the mating success is determined by the mating bias allele. So if they have the same mating bias allele, they'll always mate 100% of the time. If not, then there's a parameter we developed called alpha that'll determine the mating bias success. Yeah, so they'll go through a number of matching rounds, and after a certain number of generations, we can introduce a chromosomal inversion into the system and then proceed again with the simulation to see how the chromosomal inversion affects reproductive isolation. Okay, and so this is a way that the app interface looks. So main takeaway here is on the left-hand side, we have a face portrait, and what we're looking for is strong reproductive isolation. And that'll be determined by the face portrait, the fixed point of the face portrait converging in the bottom right-hand corner and the top left-hand corner. That determines the number of pure genotypes and what percentage of them have the different mating bias allele. With this app, we're also able to track the rate that the chromosomal inversion would invade a population, which is shown here by this line graph. And then we can keep track of the distributions, genotype distribution for the population at the beginning, before the chromosomal inversion invades, and after the chromosomal inversion invades. Okay, and so going on to some of the results from the model. So first, we wanted to see what happens when we increase the number of ecological loci before introducing viable hybrids. And this confirmed a previous finding that we had, which was as you increase the number of ecological loci, it strengthens reproductive isolation. That's due to the fact that in this case, hybrids would still be considered maladaptive. And so as you increase the number of loci, you have more hybrids that are produced. So they are eliminated due to the ecological disruptive selection. And as that continues, the disruptive selection intensifies and leads to stronger reproductive isolation. So what we have here is two simulations. The one on the left is a two loci simulation, and you can see that the fixed point shifts towards the bottom right-hand corner, signifying stronger reproductive isolation once we go to a four loci system, with all other parameters being held constant. Okay, the next result that we found from the model was what happens when we introduce the presence of viable hybrids. And what we found was that this weakens reproductive isolation, which was expected due to the increase in gene flow. And again, the weaker reproductive isolation is shown by the shift away from the corners of the fixed point when we go from a system that does not allow for viable hybrids to one that does allow for viable hybrids. So another key finding was what happens when a chromosomal inversion only captures the ecological alleles. And what we found is that negative coupling occurs. So the chromosomal inversion is able to quickly invade the system due to a fitness advantage, but that ends up disrupting the ecological selection process needed to maintain reproductive isolation. And so as these fewer hybrids are produced within the niches, sexual selection ends up undermining the ecological divergence. And what we can see here is that the system seems to converge on one of the mating bias alleles. So in this case, we have a system that captures all of the ecologically adaptive alleles. So it captures the AAA allele, and this green dot here signifies the start before the chromosomal inversion invades. And as we move towards the diagonal, we see that we lose the X mating bias allele, and it's also representative here in the bar graph. So at the beginning, the A niche has a fair amount of the X allele, but as the chromosomal inversion continues to invade, it converges on the Y allele. Yeah. Next, we wanted to see what happens when we are able to pair a chromosomal inversion that captures both an ecological and a mating bias allele. What we found is in this case, it can strengthen reproductive isolation. So this is due to the fact that the mating bias sort of has a hitchhiking effect. And so now the chromosomal inversion has a shared selective advantage where it can invade different ecological niches, and it also has paired with the mating bias allele. So what we also found was that as the chromosomal inversion is able to capture more ecological alleles, it also leads to a faster invasion rate, which is shown here on the right-hand side. Although when just one ecological allele is captured with the mating bias allele, it leads to complete fixation. So again, we have the green dot before the chromosome inversion in the single line phase portrait, and it reaches all the way to the bottom right-hand corner. When you have more than one captured, it leads to almost complete fixation, but nonetheless stronger reproductive isolation. Next, we'll move on to the mating bias model. So in this model, each individual has a multiple mating bias loci, and they can take a value of X or Y. In this model, each individual belongs to one of two niches, either A or B, and the individuals will randomly encounter one another, and their mating bias is determined by a mating bias matrix. So if they have the same genotype, they'll mate again 100% of the time. If they have a different genotype, then their mating success decreases linearly away from the diagonal, depending on how similar their genotype is. Yeah, and so they will go through a number of matching rounds, a number of generations, and then again, we can introduce a chromosomal inversion and see how the system progresses after the chromosomal inversion is introduced to the system. Again, really quick, this is just another picture of the app interface for the mating bias model. Again, we're looking for strong reproductive isolation. So we want the system to converge on the corners, the top left and the bottom right-hand corner. It's a little hard to see in this graph, but you can see the effect that the chromosomal inversion has on a system. It moves from this like green dot to the red dot in the all lines form of the phase portrait. And then this graph here, the one in the middle, shows the invasion rate of the chromosomal inversion. So in this model, A chromosome 1 version can be introduced to either of the two niches. And then again, we have a bar plot that shows the distribution of the genotypes. In this case, the uh, blue represents the individuals that belong to the A niche. The red represents the individuals that belong to the B niche. So moving on to the results for our multi-locus system. So what we found was that as you increase the number of mating bias loci, you end up having a more viable hybrid production. And as you increase the hybrid genotypes, this reduces the reproductive isolation. So in a sympatric population, what we would want is fewer mating bias loci. And yeah, when we have a chromosome inversion introduced into the system, what we found is as it captures more high mating bias alleles, it'll have a stronger fitness advantage. And the stronger fitness advantage leads to effectively reduces the number of loci, since the chromosome inversion is suppressing the effects of recombination. And the fewer hybrids will then lead to stronger pre-mating reproductive isolation. So one way we showed that here is we have two bar graphs from two different simulations. The one at the top is where 2 of 3 mating bias loci are captured, and the one in the bottom is where all mating bias loci are captured. So you can see that the one in the bottom, all of the hybrids end up being eliminated. The light blue and the light pink mean that these are individuals that have the inverted regions. The dark red, the dark blue are the individuals that have not undergone that don't contain an inverted region. Yeah. So to summarize our model, yeah, we developed a model that can factor in any number of ecological or mating bias loci. We ended up confirming previous findings that increasing the number of ecological loci or decreasing the number of mating bias loci facilitates the emergence of pre-mating reproductive isolation. Presence of viable hybrids gives rise to chromosomal polymorphism. That was something I didn't touch on. I think I was like rushing through. But when we have these hybrids and they're not affected by the chromosome inversion, they continue to resupply the hybrid niches. And that way, so in the ecological model, you can't really have complete elimination of those hybrid niches. But yeah, the chromosomal inversion is also able to capture the ones that only capture ecological alleles, leads to a decrease in reproductive isolation through negative coupling. When a chromosome inversion is paired with an ecological and mating bias allele, we have stronger reproductive isolation through positive coupling. And a chromosome inversion that captures high mating bias alleles leads to fewer hybrid genotypes and stronger reproductive isolation. Some of the limitations of our model is that it assumes all loci are fully linked. It doesn't model crossover events. There are only 2 niches and binary alleles systems, and we would need some empirical validation in order to validate the inversion dynamics and the hybrid structures. So yeah, I want to thank everyone in our lab and everyone who collaborated on this project. And with that, I can open it up for any questions. Thank you for your time.
All right. So I'm going to be presenting today on specifically coupling of habitat preference barriers and how it leads to reproductive isolation in sympatric speciation. So the question we mostly focused on was what role does habitat preference play in initiating pre-mating barriers and coupling with other barrier mechanisms to establish robust reproductive isolation in sympatric speciation. So to give you guys a brief introduction on how we began this project, we investigated how habitat preference barriers in 2 spatial configurations based on existing examples of sympatric speciation. So the first one, the open space system, is based on a population of cichlid fish which underwent sympatric speciation in the open lake. And our second one, the no open space system, is configured based on the hawthorn and apple maggot flies example that speciated over time. And to give you some more background on what an open space and no open space system in our configuration means. So here we have a mathematical model that we created on the open space and no open space systems. So for the open space model, AH represents the homebound individuals that remain and mate within niche A, and likewise for BH as well. So for the AF and BF, which are the free roaming individuals from their respective niches, they're able to swim out into the open space, and which is represented by this red line, and mate. So notice that they can also mate with their native niches, but cannot swim into the opposing niche. And the no open space model shows AH and BH as the respective niches, and FF represents the free roaming individuals from both niche A and niche B as they are able to move out to each niche. So in the no open space model, the mating occurs only at niche A or niche B, not in between. So what we found is that from the no open space system, it allows for more free movement and is still able to lead to faster and stronger reproductive isolation in compared to the other model. And in the no open space model, this is partially why the reproductive isolation is a little weaker, is because of the homebound individuals that are still being able to be visited by the free roaming individuals from each niche. So onto our methodology on how we designed all of this. So our habitat preference model, which is shown here, is broken down into more specific mathematical models, which were used to simulate populations in which we introduced the habitat preference barrier and examined for reproductive isolation. So we used our study used MATLAB and its app designer tool to generate user-friendly graphical user interfaces that solve the mathematical models of habitat preference barriers and their coupling with the two allele mating bias barrier from a previous study. If you're interested in learning more about what our mathematical models are and how the computer simulation started with the programming, here's a QR code linking to a previous video that our team has presented in the past. So here are some key takeaways that I'm going to discuss that we discovered from results in regards to habitat preference establishment, one allele versus two allele, coupling facilitation, and invasion resistance pattern. So for our first point, we found that disruptive ecological selection and maladaptive hybrid loss are needed for habitat preference establishment. What this means is, so if we look at our key variables, the offspring return ratio F, which represents the strength of the disruptive ecological selection and determines the extent of how much hybrids we lose, which is also represented by HR, is available to which is needed to drive the invasion and the coupling of habitat preference barriers. And our second variable data represents the strength of the habitat preference barrier, and it specifically specifies how effectively it reduces maladaptive hybrid loss. So to break down this phase portrait even more, the blue dots are the starting generation and the red dots are where the generation ends up. So in this case, we ran 200 generations in here. And the diagonal line represents that the ratio of alleles in this case is equal in both niches. So because there's no maladaptive hybrid loss represented by the hybrid ratio, which equals 0, habitat preference barrier in this case is unable to invade and is unable to establish reproductive isolation. And then to move on to give you some background on what a one allele versus two allele model is. A one allele model focuses on a single mutant allele that produces the same effect across different niches. So we only introduce one allele. So when the hybrid ratio is greater than 0, the one allele habitat preference barrier is able to invade and move towards fixation, and it's unaffected by recombination that we found which can be caused by gene flow. So here's a phase portrait of what a one allele model looks like. So as you can see, these starting generations are all moving towards a fixed point established by this one allele. And in the two allele model, there's two different alleles that exert effects on different ecotypes. So the two allele model tends to converge towards a fixed point polymorphism, which is more difficult to establish due to recombination. So that means within what polymorphism means in this case is that the proportion of the functional alleles exerting the habitat preference effect. So we're introducing two habitat preference alleles and its effects on each niche is interacting with each other, which makes the overall effect lower. So everyone in niche A has a K allele habitat preference, which is impacting them. And for B, it would be the J allele. So as a result, what we found is that from one allele is easier to evolve and produces a stronger reproductive isolation just because it moves towards fixation, that polymorphism. And our third point is that we found that habitat preference can facilitate the coupling of mating bias barriers. So if we look at this phase portrait, the initial generations are moving towards the center line. And this is a current two allele mating bias barrier that is divergent, meaning that it's unable to produce a fixed point polymorphism, and unable to produce a pre-mating reproductive isolation. However, if we reduce the data to from zero point 0.9 to zero point 0.6, which means strengthening the habitat preference barrier, we're able to move towards a coupling polymorphism on both sides, which means that it's establishing reproductive isolation because the habitat preference barrier is limiting the gene flow and facilitates the coupling of the mating bias barrier and strengthens the overall reproductive isolation. This is mainly accomplished by suppressing the homogenizing effect of gene flow, which would otherwise inhibit the non-random assortment of mating bias, not allowing it to couple. So to move on more about that point, so we just discussed the mechanism, which is we are able to couple mating bias and habitat preference. But what happens if you further restrict the gene flow? We notice that an invasion resistance pattern will appear as we try to strengthen the restriction. So as we decreased a number, so from earlier we had this one where it was a divergence and it converged into a single two single polymorphism where it's able to establish reproductive isolation. But as we decrease the number of matching rounds, so from 10 to 4, it creates it creates the emergence of an invasion resistance pattern. So our AX mutant mating bias allele right here, 0.01, is unable to invade into the system and moves towards 0, meaning that it can't establish into the system. So you might wonder, why does this happen? How shouldn't it help? So there's a balance between, we notice that there's a balance between a mating bias alleles, fitness advantage within interniche mating, so mating between niches, and its fitness disadvantage with intraniche mating, which is caused by sexual selection. As a result, so intraniche sexual selection is more likely to eliminate smaller populations of high mating bias mutants, which is why we end up finding a resistance pattern. So to combat that, we found that invasion resistance patterns can be usually eliminated by increasing the number of mating rounds and decreasing the number of niche sites. So since we just went over a mating round, I decided best way to show is the niche size. So here was earlier what we have from the invasion resistance pattern. So if we decrease the niche size from 0.6 to 0.1, there's like a slight little zone here that shows up where there's a space where the mutant bias allele is able to invade. And as it's shown, so we increase the mutant bias allele to AX equals 0.2, which is able to then invade and actually move towards a fixed point polymorphism. So it was actually able to establish a pre-mating reproductive isolation, whereas it was initially blocked by doing so before we decreased the niche size. So to move on to our coupling data, as we tested the amount, if we could couple amongst multiple different barrier models. If you would like to know more about what our specific barrier models mean, here's a link to, here's a QR code to the program list and descriptions if you would like to look over what each of these barrier models are described as. But we wanted to check if we noticed that one specific one couldn't couple. So why didn't this one couple specifically? If you notice here, there's a hybrid ratio that equals 0. So as long as there's no hybrid loss, the habitat preference was never able to establish in the first place. So it wasn't able to facilitate any coupling. So after these generations, there was no barrier being established. So you might wonder, does coupling order actually matter then? Because earlier when we discussed everything, we had habitat preference established first in the system and then mating bias established second. And as we discussed earlier, once habitat preference is able to invade into the system, it's very specific. Mating bias barriers are very specific and couple under certain conditions. So it's able to couple, no problem, but it has to fulfill the requirements for mating bias to be able to couple in. Whereas we tried also coupling mating bias first and then introducing habitat preference. And although mating bias was initially more difficult to establish into the system, however, whenever we established mating bias first, habitat preference was always able to couple. So in summary, habitat preference is easier to establish and tends to actually produce a stronger reproductive isolation in open space versus no open space. And as long as we've found that there's maladaptive hybrid loss, the habitat preference barrier is always able to invade to reduce the hybrid loss to the extent allowed. And a one allele model was able to generate stronger pre-mating reproductive isolation, mostly because it wasn't affected by recombination, and actually leaves a strong compared to the two allele model, which heads towards polymorphism, it heads towards fixation. And for our fourth point, so when habitat preference barrier is our first barrier mechanism, it is able to facilitate coupling of the subsequent mating bias barrier by decreasing the inter-niche encounters and mitigating the homogenizing effects of gene flow, which prevents the actual assortment of mating bias alleles. And on to our last point, we noticed that because it is so easy to evolve, the habitat preference barrier is ideally suited to establish as the first pre-mating barrier because it's actually able to encourage coupling of mating bias barrier easier to introduce and produce better reproductive isolation. And here is a picture of all our labs members and collaborators. Some of my lab members are presenting today, and you'll be looking forward to some of the other presentations. And if you have any questions, please let me know. Our preprint is actually coming out and it's going to be uploaded to archive in a month. So if you have any questions about it and you want to look at more data about what we did, you can look on that. And I'm ready for any questions if anyone has any.
Today, I will be presenting on one of our papers entitled Computer Simulations and Analysis of Coupling Among Reproductive Barriers in Late-Stage Sympatric Speciation. My name is Lauren, by the way, and a couple of my lab partners, Karen and David, also presented earlier, so some of this information may be familiar or some of the modeling might be familiar, but let's get started. So in a previous study, we developed a mathematical model and derived this two mating bias allele, two ecological niche model from this previous study. We also used individual-based computer simulations to propose this 5-stage process for establishing initial pre-mating reproductive isolation in sympatric speciation. So the aim of this study is to take the same model and these same computer simulations to investigate coupling of additional reproductive barriers during later stages of speciation. Some important things to note here, just some variables that are going to continue to come up. The variable alpha here changes mating biases and affects pre-mating reproductive isolation between ecotypes, whereas variable F changes hybrid viability and post-mating reproductive isolation. So in this last diagram, figure M1 at the bottom shows the life cycle of a sympatric population used in our computer simulations. So in each generation, individuals in the population undergo ecological selection and then sexual selection to produce offspring for the next generation. Here's an example of the computer model we developed to solve the set of nonlinear difference equations from our mathematical models. It should look familiar from a couple of the previous presentations, but you can see all of the relevant variables here. For example, that variable F and alpha mentioned earlier. All of the parameters here are normalized, and in the phase portrait, you can see we plot AX versus BX, AX being the ratio of individuals in niche A with the X allele, and same for niche B. So as we change the parameters here, we can see how the phase portrait changes. The blue dots are the initial starting populations, and here after 100 generations, you can see how the population ratios of AX and BX converge to a fixed point polymorphism shown by the red dot. So as they move closer to the bottom right hand and top left corner of the graph. So getting more into this paper, this diagram really represents the central focus of our paper and what our results showed. We analyzed 5 barrier mechanisms, 2 in this early stage, so mating bias barriers and habitat preference barriers, and 3 designated in late stage barrier mechanisms. So that's chromosomal inversions, mutations reducing hybrid viability, and BDM incompatibilities. So what we found was that these 5 barrier mechanisms work in a positive feedback loop, which I'll definitely get to later. But first, we're going to look at how these mechanisms act individually. Starting with the early stage mechanisms. So as I mentioned, the 2 early stage mechanisms will be habitat preference and mating bias barriers. And these, both of these barrier mechanisms we found to be adaptive So driven by their selective pressures created by hybrid loss. Both of these mating bias and habitat preference barriers can independently establish initial barriers because of this, which can then couple with other similar adaptive pre-mating barrier mechanisms to progressively increase pre-mating reproductive isolation and further reduce gene flow. As mentioned earlier by Karen, because habitat preference barriers are more readily established than the two allele mating bias barrier, establishing habitat preference first and then coupling it with mating bias, it presents a possible pathway for initiating pre-mating reproductive isolation in sympatric speciation. But also something to note is both of these are much more easily reversible. So let's go ahead and look at some of the results. So we ran these computer simulations across a bunch of different combinations of these barrier mechanisms. So these are just a couple of examples. This first one is coupling of initial two allele pre-mating barrier mechanism with a one allele barrier mechanism. And the second one is a two allele pre-mating barrier with another two allele barrier mechanism. And so in these diagrams, you can see again that the red dot moves closer to the bottom right-hand corner as the fixed point polymorphism moves towards fixation in the niches, representing stronger pre-mating reproductive isolation. So kind of coming back to this, we're going to dive into the late stage barrier mechanisms, and those are a little more complex, so I'm going to spend a little bit more time on them. These are our 3 late stage barrier mechanisms that we analyzed. They are adaptive and non-adaptive, depending on which one we're talking about, and we'll get to that. They do occur in pre and post mating, affect pre and post mating reproductive isolation, as opposed to the early stage mechanisms, which only affect pre-mating reproductive isolation. And they are less easily reversed than the early stages. So first, mutations reducing hybrid viability. So these mutations are late stage as they are underdominant, so they're affected by gene flow. In the early stage, you have a lot of interniche mating and increased gene flow, and in the late stage, you have less gene flow, which causes this a smaller fitness disadvantage for these mutations reducing hybrid viability. They are adaptive as your fitness advantage is controlled by gene flow. For example, a mutant allele that increases hybrid incompatibility and decreases hybrid viability can't invade by itself, has a difficult time invading by itself because individuals carrying the allele are at a fitness disadvantage. However, when coupled with an allele that has an invasion advantage, whether it's through physical linkage or chromosomal inversion, the mutant allele can hitchhike with the advantageous allele and invade. So this can combine with chromosomal inversions and or BDM incompatibilities in order to gain this net fitness advantage necessary to invade. After the mutant allele is able to invade and rise to near fixation, even if disruptive ecological selection is reduced, the intrinsic hybrid incompatibility conveyed by the mutant alleles can still prevail to ensure continued hybrid loss and maintain that established fixed point polymorphism and pre-mating reproductive isolation from earlier so that the pre-mating mechanisms don't collapse. Moving on to the next one we're going to look at is chromosomal inversions. I'll keep really brief because we did see a presentation on this earlier from David, but chromosomal inversions are important as they're another late stage barrier mechanisms. They are dependent on non-random assortment. So when you have non-random assortment, obviously reduced gene flow, it's easier to capture favorable alleles, and they are adaptive. So these linked alleles, or we call them super gene in our paper, they can't be broken up, so they're advantageous compared to other individuals to invade. There is a gene flow intermediate range, so gene flow too high, you can't capture locally adaptive alleles, and when the gene flow is too low, there's not enough hybrid loss. We do have this issue of negative coupling, where if you capture only ecological adaptive alleles, this decreases ecological selection against hybrids necessary for pre-mating reproductive isolation. So you do need to capture both ecological and mating bias alleles in order to prevent this negative coupling. And here I just included another example from our results section of a two-allele premating barrier with chromosomal inversion. All right, moving on to the last one is going to be our BDM incompatibilities. So this is a late-stage mechanism. It operates best under low gene flow. It is non-adaptive, so as opposed to the first two late-stage mechanisms that I talked about, BDM is not driven by hybrid loss, so it's non-adaptive. It's an incidental byproduct of ecological adaptations. It is the slowest mechanism of all of the ones that we looked at, so it evolves the slowest, but it is the most irreversible. So in the final stages of reproductive isolation, when gene flow is very low, BDM incompatibilities we found can evolve independently as the genome continues to diverge, and makes this very difficult to reverse because they're all independent of each other. And here's another example of from our results section of coupling an initial two-allele premating barrier with a one gene locus BDM barrier mechanism. This I just included. It's part of our results section as well. It's in our paper. If I'll have a link, a QR code to it at the end. But this kind of discusses in more terms what I went over and the differences we found between how these barrier mechanisms act in early stage and late stage, and also the similarities. So finally, I did mention briefly at the beginning this positive feedback loop. So now that we've kind of dived into how the differences between these barrier mechanisms, we looked at how they all interacted with each other. And what we found was that the early stage mechanisms establish initial premating isolation barriers, so this initial premating reproductive isolation, and those barriers kind of chokehold and reduce gene flow, which facilitates late stage barriers that are dependent on that reduced gene flow. Then the late stage barriers can strengthen, produce post-zygotic reproductive isolation, and further reduce gene flow. And then they also secure those early stage mechanisms by with intrinsic hybrid loss to sustain premating reproductive isolation, even if disruptive ecological selection does decrease. The order that barriers are coupled does matter because coupling barriers becomes more difficult as the reproductive isolation strengthens. And as I mentioned with the BDM incompatibilities, at the very last stages, when there's very little gene flow, many different post-zygotic mutations and barriers can evolve independently of each other, making it difficult to reverse. So in conclusion, we extended a previous mathematical model to demonstrate how an initial barrier can facilitate coupling of additional barrier mechanisms. We used computer simulations and analyses to investigate coupling among different reproductive barriers in sympatric speciation. Our results showed that after initial mating bias barriers reduce gene flow, it can facilitate the coupling of other barriers to produce stronger and irreversible reproductive isolation. And we saw that this works in that positive feedback loop that I kept bringing up between the early and late stage mechanisms until you have stronger and stronger reproductive isolation. And so together, the results of our two studies, that first one that we keep mentioning with, where we developed this mathematical model, and the this study now where we're kind of looking at the overall possible combinations of different barrier mechanisms, outline a possible pathway forward for the progression of sympatric speciation. While our proposed mechanism still requires empirical validation and more theoretical refinement, we are just trying to offer a valuable framework that could be improved and extended through future research and discussions to advance our understanding of sympatric speciation. And with that, there we go. Oh, sorry. This is the QR code to our preprint. Excuse me, if you have any questions at all. There's obviously a lot more information in there, and I was kind of trying to move through it as quickly as possible. And I want to thank all of our lab members and collaborators and take any questions at this time.